Hi everyone, this is lecture 22 on nutrient digestion and absorption. This is the third lecture on the digestive system. And in this lecture, we're going to talk about how the different nutrients are actually uh, mechanically and chemically digested and absorbed. Because remember, until it's absorbed, these nutrients that you obtain from your diet are not really part of you yet. So we're going to talk about the enzymatic digestion of carbohydrates, lipids, and proteins, as well as briefly about nucleic acids. And we're going to talk about how they're absorbed across the small intestine. So it is the small intestine that's where the majority of the absorption occurs. Okay. In addition, vitamins, water, and electrolytes are also absorbed in the small intestine. So let's start from the beginning and talk a general overview of digestion and absorption. Okay, essentially digestion is breaking down the food so into smaller particles, and so that we can bring it into the body through absorption. There are two types of digestion: mechanical digestion and chemical digestion. Okay, mechanical digestion is the physical breaking of food into smaller parts. Okay, so it involves such processes such as mastication or chewing, churning, the movement of the muscles of the intestine and the stomach to break larger particles into smaller particles, and also segmentation, which is another type of peristalsis. Chemical digestion is the breaking the chemical bonds of food molecules. So this requires the enzymatic digestion. It varies for each nutrient because digestive enzymes are specific for a single type of bond. You learned in 203 that enzymes are fairly specific. However, if you remember from SAB 203, one of the major reactions of breaking these bonds is through a hydrolysis reaction. Now, if you remember, a hydrolysis reaction is a type of reaction which uses water to break the bonds between two molecules. And of course, water is prevalent in the digestive system because it's present in large amounts in gastric and pancreatic juices. So just to give you a reminder, or if you had forgotten, here's an example of a hydrolysis reaction. Here we're using a protein as an example. Here's a dipeptide. Here's a peptide bond. Remember, a dipeptide is a molecule with two amino acids. Water is a reactant, which is going to break the peptide bond. And in the process, water becomes part of each of the individual amino acids that are produced. So the hydroxyl group, the OH, is added to the carboxylic acid group, and the hydrogen is added to the amino group. And so, as a result, we have two separate amino acids through this hydrolysis reaction. And we use one molecule of water is consumed in the reaction. Now, once digested, the nutrients must be absorbed across the epithelial lining of the alimentary canal. Okay, so remember, they have to cross both the apical membrane, and then they have to cross the cytoplasm and out through the basal membrane into the interstitial fluid, and then into the blood or maybe lymphatic system for delivery to the body cells. Okay, so this figure here shows a summary of what we're going to be talking about for each of the specific molecules that we absorb. Okay, so a molecule must cross the apical membrane. So here's the lumen of the small intestine. The molecule must cross the apical membrane of the enterocyte, plasma membrane, via channel or transport protein. Now it's in the cytoplasm. It has to diffuse and travel across through the cytosol, and then it must exit the basal side of the plasma membrane into the interstitial fluid. And here in this picture, we show this molecule entering the into the bloodstream through a capillary, and then it's taken to the other parts of the body. So when we talk about digestion absorption, we're going to talk about the major organic molecules that you learned in SCP 203, such as carbohydrates, proteins, and lipids. We'll also talk a small bit about nucleic acids to finish it off, since we learned all four in SCP 203. Let's begin with carbohydrates. So the digestion and absorption of carbohydrates. Okay, so for all of these molecules, you want to know where actually digestion occurs. Okay, so for carbohydrates, it begins in the mouth with an enzyme called salivary amylase. Okay, and is completed in the small intestine by an enzyme called pancreatic amylase. Okay, so the pancreatic secretions enter into the duodenum of the small intestine. So digestion of carbohydrates occur both in two places, the mouth and in the small intestine. Okay, so digestion begins of carbohydrates in the mouth, where long polysaccharides are broken down to shorter oligosaccharides. It's incomplete because there's not enough time. However, if you eat a cracker, for example, and you chew it long enough without swallowing, you should feel a sensation of it being sweeter as the carbohydrates are broken down further. But generally, we wolf our food down, and so we don't really notice that salivary amylase working too well. So do you think salivary amylase continues to work after it reaches the stomach? Well, if you remember the nature of the stomach, we would say no, because remember, the stomach is a very harsh environment. It's very acidic with HCl, and so the proteins are going to get denatured by the acidic pH of the stomach. And so once the salivary amylase is swallowed with the carbohydrates and the rest of the food, it becomes inactivated once it hits the stomach. And of course, the time in the esophagus is so short that there's not enough time for anything to happen there. However, in the small intestine, we have another enzyme called pancreatic amylase, where it digests it further. And so pancreatic amylase will digest the polysaccharides further into oligosaccharides in the small intestine. So in this first figure here, we see here's the lumen of the small intestine, and here's the epithelial lining of the small intestine. We see that polysaccharides are broken into oligo and disaccharides in reactions catalyzed by pancreatic amylase. So here's the polysaccharides. They're broken down to oligosaccharides and disaccharides. Now, do you remember the brush border? Okay. Well, the brush border has a large surface area, so it contains many enzymes along the apical surface of the enterocytes. And these brush border enzymes will complete the digestion of the oligosaccharides. So we have such enzymes such as lactase, maltase, and sucrase that will further break down these oligosaccharides. Okay, so here we see these oligosaccharides that are broken down to disaccharides and monosaccharides by these brush border enzymes. Okay, once digested into monosaccharides, they are now absorbed. And they have different ways depending on the type of simple sugar. Glucose and galactose move against their concentration gradients by using a sodium-glucose cotransporters, which you might have learned in SCP-203. Remember, a cotransporter operates by moving two or more molecules in the same direction. So both glucose is brought in through the driving force of the sodium. Okay, remember, extracellular sodium is higher than intracellular sodium. So by using this concentration gradient, glucose is brought into the cell. So here we see that the movement of sodium, the driving force of sodium from high to low, is coupled with the movement of glucose. So wherever sodium ion that's moved in, one molecule of glucose is brought in. And this is an example of secondary active transport. Okay, it's called type of secondary active transport because it's using the driving force of the sodium into the cell. However, the movement of the sodium has to be moved out using the sodium potassium ATPase. Okay, and so the sodium pump is using ATP to drive sodium out of the enterocyte to maintain that sodium gradient into the lumen of the small intestine. Okay. So since it's going against its concentration gradient, this is a primary active transport process and uses ATP. So this occurs both for glucose and galactose into the enterocyte. So these are both secondary active transport processes. Okay, so here in this figure here, we see the sodium-potassium pump, which creates the gradient for the sodium absorption out here. Okay. Now, fructose, on the other hand, uses facilitated diffusion. Okay, and it uses a transporter here that moves fructose from the lumen into the enterocyte. Okay. So secondary active transport for glucose and galactose facilitate diffusion by fructose. Now, once they're inside the cell, the movement of glucose, galactose, and fructose across the use facilitate diffusion across the basal enterocyte membrane. Then they diffuse into the extracellular fluid, and then into the villus capillaries, and then into the liver for processing. Okay, so here they use facilitated diffusion into the interstitial fluid and into the blood capillary, and they're off on their way to get to the liver to be metabolized, so the cells can use it. Okay, let's look at the digestion and absorption of proteins. These begins in the stomach and is completed in the small intestine. Okay, so this is to the two different sites, but they're in different locations than in the carbohydrates. Remember, chief cells produce the inactive pepsinogen. Okay, and then pepsinogen becomes activated by the low pH in the stomach. And the inactive pepsinogen is converted to the active pepsin. What's interesting is that its optimal pH is at low pH. If the pH goes to pH 7, pepsin becomes inactivated. So it has a very limited range of activity, but it's ideal for the stomach. So pepsin digests about 10-15% of the ingested proteins into smaller polypeptides, oligopeptides, and free amino acids. So not a significant amount occurs in the stomach. The remainder of the protein digestion is in the small intestine. So we see that the small intestine is very active in digestion with pancreatic and breast border enzymes. And there are 5 pancreatic enzymes that digest proteins. And they're all released as inactive precursors. Okay? So you might ask, why are pancreatic enzymes released as inactive precursors? Well, there's a very logical explanation for that. Because it prevents the pancreas from digesting itself. So that they're secreted as inactive precursors, and then they can be activated outside the cell. Now, there are several pancreatic proteases, and there are many more than what we're naming. So we're just going to give you an example of some of them. The first enzyme is trypsinogen, and so whenever you see this suffix, it's generally going to be an inactive enzyme or inactive protein. Okay, so trypsinogen is inactive, which becomes the active trypsin. Okay, and trypsin then can activate other pancreatic enzymes, even trypsin itself, so that as trypsin is formed, it can activate other enzymes as well as trypsinogen, and so now they're active to digest proteins. Other pancreatic proteases which digest proteins include chymotrypsin, carboxypeptidase, dipeptidase, and tripeptidase. So some of these, such as chymotrypsins, have very specific cutting specificities between certain amino acids. Others are less specific, such as carboxypeptidase will cleave at the C-terminal end of a protein. Okay, dipeptidase will cut dipeptides, and tripeptidase will cut tripeptides. And then finally, brush border proteases will digest the oligopeptides into free amino acids. Okay, and so remember the brush border is right along the membrane, so they're perfectly suited so that once they're digested, then they can be absorbed. The free amino acids and some oligopeptides are then absorbed. Okay, so here this diagram is showing the oligopeptides, which are broken down into free amino acids, and there might be some, and they're digested by these brush border proteases. And they also use the sodium gradient from the sodium potassium pump to cross the apical enterocyte membrane. So just like glucose and galactose, a secondary active transport process using the driving force of sodium will bring amino acids into the cell. So this is a secondary active co-transporter. And they're using the sodium driving force from the sodium potassium pump. Now, oligopeptides and free amino acids cross the apical membrane by secondary active transport. And then once inside the cell, the remaining oligopeptides that are brought in are digested into free amino acids in the cytoplasm. Okay? The free amino acids exit the basal membrane by facilitated diffusion, and then they enter the villus capillaris and to the liver via the hepatic portal vein. Okay? So here they leave by facilitated diffusion into the interstitial fluid and into the bloodstream and on their way to the liver. Let's look at the digestion and absorption of lipids. Okay, in this case, lipids are a little more complicated, mainly because they're hydrophobic. And most of the dietary lipids are going to be triglycerides. Okay, lipids are broken up two ways: through mechanical digestion, through mastication, churning, and segmentation, and also by a special process called emulsification. Okay, by amphipathic bile salts in the small intestine. Okay, so remember bile salts were produced by the liver and stored in the gallbladder. Now, do you remember what amphipathic means? Well, amphipathic molecules are those molecules that have both a hydrophobic part and a hydrophilic part. So amphipathic molecules have both. Okay, the hydrophobic part of bile salts are derived from cholesterol by the characteristic 4 rings. Okay. So bile salts are amphipathic because they have both a hydrophilic end and hydrophobic end. The nonpolar or hydrophobic end interacts with the lipids. That makes sense. The hydrophilic or polar part interacts with the water. So bile salts sort of acts sort of like a detergent in a way. And by doing this, it breaks up big globules of lipids by a process called emulsification, so that it pulls apart and breaks up these large globules into smaller ones. And the idea is that by breaking them into smaller globules, there's more surface area for the lipases to act on. So true or false? Bile salts digest lipids. That's false. Bile salts emulsify or break up lipids, forming an emulsion into an emulsion. An emulsion are tiny lipid droplets coated with bile salts. Okay, when we say digest, we refer to that chemical bonds are broken through enzymes. Okay, so bile salts don't digest; they emulsify. About 15% of the lipids are broken up by the stomach churning and gastric lipase. Okay, so not a significant amount due to the large size of the globules of fats that does not have a very high surface area for the lipase to act on. The way gastric lipase works is it removes one fatty acid from triglycerides. Remember, triglycerides is glycerol with 3 fatty acids. If it removes one fatty acid, then you end up with a diglyceride and a free fatty acid. So that's the action of gastric lipase. Okay? So in the stomach, lipids are broken down by the gastric lipase. Okay, so we end up with diglycerides and the free fatty acid. Okay, by the gastric lipase. Lipids enter the small intestine and then are emulsified by bile salts. And remember, emulsification is only a physical process. No bonds are broken. All they're doing is breaking big globules into small globules by bile salts. And so the bile salts pulls apart these large globules, separating them in the aqueous media of the gastric juice. Then once in the small intestine, pancreatic lipase digests lipids into free fatty acids and monoglycerides, because now they're separated by the bile salts and there's sufficient surface area for these enzymes to act more efficiently than gastric lipase. So here we can see that due to pancreatic lipase can act on more efficiently these emulsified fats into monoglycerides and free fatty acids. Now, micelles are the bile salts that remain associated with the digested lipids, and they remain associated as micelles. Reason why is that the micelles prevent lipids from reforming back into large globules. That kind of defeats the purpose of breaking them up. Okay, so finally we see that the bile salts remain associated with the digested lipids in the form of micelles. And so here's a micelle here. So let's look at the summary of carbohydrate protein lipid digestion. So you're going to have to know the sites and processes where each occur. Okay, and so here's the summary that kind of shows the differences between carbohydrates, proteins, and lipids. Okay. Let's continue looking at lipid absorption. Okay. Now, just as lipid digestion is more complicated than protein and carbohydrate digestion, lipid absorption is also complicated. Okay. Let's continue on with the micelles. The micelles take lipids to the enterocyte plasma membrane. Okay. And these micelles contain not only triglycerides, but also other products of other fats, such as cholesterol and fat-soluble vitamins. Since they're hydrophobic, they can diffuse through the phospholipid bilayer by simple diffusion. So they don't need any kind of transport mechanism. So here's the micelles, okay, and then now they're still attached to bile salts, but now that the lipids can diffuse right through the membrane since they're hydrophobic. Inside the enterocytes, lipids reform back into triglycerides. Okay, this seems to me kind of a little kind of backwards, because we broke down the triglycerides for them to be reabsorbed, and then once they're inside the enterocytes, they reform back into triglycerides. Well, they can do this now because they don't have the bile salts attached to them anymore. These are packaged into the chylomicrons in the Golgi and exit the basal membrane by exocytosis. Okay, the chylomicrons enter the interstitial fluid and then enter a lacteal. Okay, remember the lacteals are part of the lymphatic system that will help absorb fats that are too large to enter the circulatory system. So in this picture here, we see the monoglycerides and fatty acids reform as triglycerides, and in the Golgi apparatus, they're packaged in the form of a chylomicron. These chylomicrons then are transported to the basal membrane and leave the cell by exocytosis, and then can diffuse into the lacteals. The empty chylomicrons now are taken apart in the liver, and the hepatic portal vein returns bile salts to the liver to make new bile. Okay, and of course the lipids will eventually return back into the circulatory system. That part you learn in SCB 203. Finally, let's look at the digestion and absorption of nucleic acids just a bit, so that we finish off all the organic molecules. And the enzymes that digest nucleic acid are called nucleases. Okay? The digestion of nucleic acids occurs on the brush border brush border. Which is a little typo here. It should be here. So the brush border nucleases removes the phosphate and the sugar from nucleic acids. Remember, nucleic acids are composed of nucleotides, and nucleotides are composed of a phosphate, a sugar, and a nitrogenous base. Okay, so the phosphate, sugar, and nitrogenous bases are absorbed by the villi capillaries, and the hepatic portal system takes them to the liver to be metabolized. Okay, so we didn't talk a lot about nucleic acid. So finally, we have the summary of the digestive enzymes. So this is a good review table that talks about all the enzymes we mentioned in this lecture, their source, and their actions. So you're gonna you should know these. And finally, let's look at the summary of digestion. And I put so far because we're not quite finished with the digestive system because we haven't talked about the large intestine yet. But by far, most of the digestion and absorption occurs by the small intestine. Okay, so I've circled the part that we covered so far, and of course we haven't talked about the large intestine yet, which we'll cover in the next lecture. Okay, and of course, the large intestine has very important functions as well. Okay, as we can see that it's going to have important for reabsorption of electrolytes, water, and vitamins. Okay, so that's the setup for the next lecture.